This recording is looking at Chapter 17. As I alluded to, Chapter 17 will be divided into two portions. Chapter 17 is the blood. The first aspect will have to do with more of the introduction, talking about red blood cells, white blood cells, hematopoiesis. After that time, we will actually leave this chapter and go to Chapter 20, the lymphatics. So Chapter 17 will work into what I call my cardiovascular module and my immunology module, which you can read on in D2L as far as my explanation. The first slide here has to do with water content in the body, which you should have learned in A&P 1. Water in the body is divided into two. That water inside the cell is called intracellular. That outside the cell is called extracellular. Extracellular water is divided into interstitial fluids and into, which is also called tissue fluids, and also that in blood vessels, which we call intravascular. Adult males have about 60% of water, females slightly less due to the amount of fat tissue. Again, in a 70 kilogram, which again is our standard human, about 56 to 60% of the body is water. If we equate that, that's about 40 liters or 40 kilograms of water out of the 70 kilograms. Most of the water is located inside the cell, because that particular person has close to 100 trillion cells, with some water on the outside. The plasma, around 3 liters, which is a component of the blood, and then the interstitial fluid, which is that which bathes the cells. Here again, you can look at a division of the water. Now, in my face-to-face class, I talk about how to obtain blood from a patient. One is the venipuncture, which is the most common for many reasons. One, you can obtain a lot of blood, and the flow rate is not that fast, so that infection once you push the needle in, because again, you never sterilize where you draw blood from, you just reduce the bacterial concentration. Arterial stick, the only time it's mandatory to do an arterial stick is doing something called an arterial blood gas when you're trying to determine the amount of oxygen. Most of the time, because of the high pressure on the arterial side and the fast circulation, we avoid that. Capillary stick, which is also called a finger stick, is where you obtain just a little bit of blood in order to do certain tests. But it's not a good source because you can only obtain a little bit and you have to squeeze too hard, which disturbs the blood count. The composition of blood is basically this. You have what we call the plasma portion of the blood. Now, plasma minus some of the clotting factors is what we call serum. Plasma minus some of the clotting factors is what we call serum. But the two components of blood is the plasma and the formed elements. We call them formed elements instead of cells, even though sometimes you may hear me say cells, because the only cells of the blood really are the white blood cells. They still contain their nuclei. The platelets and the red blood cells lose their nucleus, so that's why we use the term formed elements. So the formed elements would include the erythrocytes, which are the red blood cells, the leukocytes, which are the white blood cells, and the platelets. We do something called to spin an hematocrit. Spinning a hematocrit is where you draw some blood, put it in a centrifuge, and the heavier stuff, which would be the formed elements, goes to the bottom with the plasma on the top. And so what we're able to do is to determine the percentage of plasma to the formed elements. Now, red blood cells in amount are far higher than white blood cells. Red blood cells are in millions. Platelets and white blood cells are in thousands. So oftentimes in the clinical situation, when we spin an hematocrit, we're actually trying to see if the person is anemic or trying to see how many red blood cells. Because as you can see in this column, most of the cells are red blood cells. They go to the bottom. White blood cells, which are a little lighter, stay at the top, and it's a foamy, buffy coat there. So the hematocrit gives that percentage. Here, it's about 55% there on the plasma portion and 45% you can see on the formed elements. The hematocrit, though, ranges from something like about 30 to 60, looking at the formed elements. So as low as 30, as high as 60. A high hematocrit increases too many cells and causes an increase in viscosity of the blood, which can cause the blood to sludge. When blood sludges in the blood vessels, it attempts to clot. I'm not going to talk about, which I would in face-to-face, the various test tubes we draw blood in. I'm going to go to the physical characteristics of blood, which you can see. Key points here. The blood pH needs to stay in homeostatic range of a 7.35 pH or 7.45. Blood temperature is about 100.4 because most blood circulates in the core of the body. You may recall from ENP1, the core of the body is hotter than the surface. The volume of blood has a median value of about 5 liters, about 5 to 6 for males and about 4 to 5 for females, with a median for both sexes at 5. Hematocrit, as I alluded to, is about a 30 to 60. The viscosity of blood is measured in centipoise, but the key to this, don't really have to memorize that, is that the higher the viscosity, the more sludging the blood. The high viscosity would be due primarily, but not only, to an increase in the number of cells. Here again is just another chart showing you everything. Here's kind of looking at various temperatures of the body. We won't cover that. The functions of blood, as you can see, are transport, regulation, and protection. Obviously, you know that blood transports many things: gases, nutrients, all kind of hormones, enzymes, everything. Regulation, it helps, as you know, from ENP1, control body temperature. It helps. It also is important in the control of your pH of the body and your blood volume. Regulation, primarily, again, body temperature, the pH. Protection primarily has to do with hemostasis, which means to stop bleeding, and also the immune function of the blood. Here are the formed elements, again, red blood cells, white blood cells, and the platelets, which you can read on. As I alluded to, red blood cells are in millions. You can see about a 4.8 to 5.2 per microliter. You have to know the units. White blood cells about a 5,000 to 10,000 per microliter. And then the white blood cells are subsumed into cells called the granulocytes. A granule is a stained vesicle in histology. So the granulocytes have lots of granules. The neutrophils, the eosinophils, and the basophils are your granulocytes. The agranulocytes, I wouldn't have named them this. I would have called it oligogranulocytes, because it's not like they have no granules. A means without. They just have far less granules, which are your lymphocytes and your monocytes. Here again is kind of a breakdown of the relative percentages, which you can study of that. Here's some pictures of the cells as you would look under the microscope as far as a drawing. Here again is another drawing. These are white blood cells. Most of the cells you can see are red blood cells because they are in millions. And these little dotty things that you see there are really your platelets, which are about 2 to 4 micromillimeters across. Here's your blood plasma. It has a lot of solutes in it. That's the fluid part of the blood. Remember, plasma minus some of the clotting factors is what we call serum. Here's your blood plasma. It consists of about, let's say, 91 to 92% water. 7% is due to something called the plasma proteins. The plasma proteins are your albumins, your globulins. You have 3 types of globulins: the alpha, the beta, and the gamma. Gamma globulins are also called your antibodies. And then you have something called fibrinogen, which you may have heard about in ENP1. That's associated with clotting. Then you have other solutes in the blood. I'm sorry, I moved on. You have solutes in the blood. Some of those being your electrolytes would be your sodium, potassium, and chloride, and so forth. Nutrients, main nutrients would be your glucose, but there are other nutrients being transported. Gases would be like your oxygen and CO2, many hormones, many enzymes. Waste would be like urea and uric acid. Urea is breakdown of your amino acids, and uric acid is a breakdown of your nitrogenous bases like DNA and RNA. Now, I want you to study a little bit in the text on hematopoiesis, which is the formation of blood. I started mine because I have time in face-to-face as to the beginning of it all, where your sperm met egg. Basically, to say it in a simple way, sperm meets egg, if fertilization occurs, you have the first cell. The first cell, called the zygote, is a totipotential stem cell. That means that cell can form anything. Then what will happen is you will have a proliferation, mitosis, of a bunch of cells until you finally get a whole bunch of cells as you go through the 2-cell stage, 4-cell, and so forth. And then after you get to a certain number, now all of these here are just alike. So these would be called totipotential stem cells. A stem cell can do mitosis and differentiation. Then what will happen is eventually you will start to get differentiation of these cells, cell specialization. You may recall from ANP1, you had 210 different cell types. So what will happen is these cells will start to migrate within the entity and differentiate. The initial differentiation will give you, you may recall, your 3 germ layers. Your 3 germ layers. Your 3 germ layers were ectoderm, endoderm, and mesoderm. Ectoderm, endoderm, and mesoderm. And from these 3 germ layers, when you go through different things. For example, ectoderm can be involved with forming the skin and so forth. Mesoderm, blood vessels and blood, and your connective tissue, which blood is a connective tissue. Endoderm assists with the formation of the gastrointestinal tract. So you can kind of glance. Don't get very heavy on these. These are a little more than you need right there. I come here. Here would be a mesenchymal cell. You may recall from ANP1, mesenchyme is a subordinate of mesoderm. Mesenchyme cells can form adipose cells, fibroblasts, which does soft connective tissue, various things, your cartilage, your bone. But what I'm interested in is something called a hemocytoblast cell. A hemocytoblast cell is the initial blood stem cell. All of your blood-formed elements form from that. So from that you can form your red blood cells, your white blood cells, and your platelets. Okay? This slide here shows, again, don't get very detailed, where the various locations of blood are as it's being developed. Blood develops extravascularly. In other words, blood develops in blood islands, and then blood vessels grow to surround the blood. So blood develops in blood islands, and blood vessels come and encompass the blood. Here would be a chart showing the various locations that blood is being made. Again, so right here, this is a hemocytoblast, the initial stem cell for blood. And from that, you can see you form a myeloid stem cell and lymphoid. This forms the lymphocytes, and the myeloid stem cell differentiates further to form all the other blood cells. Again, here is another chart on that particular aspect. Here's kind of a look at the different developmental cells. Hematopoietic stem cell, multipotential stem cell. Again, these are things that normally I would discuss in class, but I won't get very detailed on that in this online presentation. In order for blood to develop, it has to use certain growth factors and also hormones. In other words, blood develops homeostatically, which means that you only develop blood as blood is needed. Blood is being formed and blood is being destroyed, as with everything else in the body. So in order to keep the blood counts in normal range, these growth factors come in along with hormones to keep the blood in normal range. One of the major growth factors, one of the major hormones we'll talk about is this one called erythropoietin, which is the main hormone that stimulates red blood cell. Thrombopoietin is big in the stimulation and formation of platelets. Okay, I'm going to end this discussion here, and then I will do another one on red blood cells. Thank you.